Okay, so this lesson will be over chapter 4, primarily chapter 4. If you remember with chapter 1, I skipped over a few pages that concerned classification because I wanted to start off in chapter 4 focusing on classification and also talking about staining. Now for microscopy, what I do is I have you all write a paper, and in the actual paper section is where you're going to find the actual video instructions of a little bit more details on how to do the paper in case you are still confused or in case you have any questions. I think that way me verbally going over it as well as the typed information will clarify anything that you may have as far as any concerns you may have as far as your paper goes, because I want to make sure you guys can do that and you feel like you're going to be successful. So let's go ahead and start off with no further ado with the classification section. And with that, I want to actually not start here, but what I'm going to start at is all the way back at chapter 1 in your textbook, where we start talking about how can microbes be classified, where it starts on page 4 talking about classification of microbes. Now I'm going to go ahead and start off with our types of cells, because you know that always has to be something we talk about in our classes. And there's two main types of cells, period. And those two types, you may have heard of this one definitely before. You have eukaryotes, and we also have what's called prokaryotes. Okay, to help us to understand, to me, I usually say you are a eukaryote. So that way you understand these are typically multicellular. Whereas prokaryotes are typically unicellular. I can't seem to spell today. These typically have a nucleus. That's literally what EU means. EU means with, and then karyon, which is the basis of what this is. Means nucleus. These are Latin terms. Pro, before or no, and of course karyon, nucleus. So those are two basic types that we're looking at there as far as cells go. We need to really understand that and make sure that we have a good understanding of those two types. Okay, so we've got these two cell types. I love my whiteboard. It's so awesome. If you have one, you definitely want to use it for notes. They're great. So I've got my two cell types right here. Multicellular and unicellular organisms are going to be based upon these two main cell types, eukaryotes and prokaryotes. Now, when we go into classifying these cell types, eukaryotes belong in a certain domain. Domains were created not that long ago by a man named Weiss. That was his last name. He came up with that actually in the 80s. He said, hey, all these there's two cell types, excuse me. All right. I know that and I understand that there's two cell types. However, there's some differences in these prokaryotes. Their RNA structures are a little different. So since their RNA structures are a little different, I'm going to split them up into domains so that way we can classify them accordingly. And he also understood that, hey, these were totally different types of bacteria. So we started understanding a little bit better about prokaryotic organisms, which are mostly bacteria, and we started understanding that, hey, we need to split these up a little further. So the domain for eukaryotes is eukarya, meaning with nucleus. And then we have your prokaryotes. They're going to be split into two different ones, which one is bacteria and the other one is archaea. Alright, so there's our domains and our classification structure. Now I'm kind of bouncing around here, so let me get a little more firm footing here because I don't like how I'm kind of moving around with you guys. Alright, so when I'm talking about classification, this is their domain classification structure, and that would be a category, or what we would call a taxon. Okay, a taxon is a classification category. Now, let me talk to you a little bit about classification and that information there. So I'm going to kind of bounce over here. And let me get a little bit better with my lesson here because I'm kind of bouncing everywhere. You guys are probably going, where are you going? Alright, so classification. Way back in the day, in the 1700s, right around Leeuwenhoek, we remember him, we had no way of classifying organisms in any form or fashion. And remember, honestly, we didn't know a whole lot about organisms either, because remember, Leeuwenhoek just called them animal kills, his bacteria, because he didn't have any understanding of what he was looking at other than just these are really tiny moving organisms. That's all he understood. So he called them animal kills because they moved. If they move, they gotta be an animal. If they don't move, they gotta be a plant. That was pretty much our understanding back then. And a guy by the name of Carolus Linnaeus, this is all on page 4, came around and he discovered classification. He didn't discover classification. He is the one that originally came up with classification. And I cannot spell today. I'm sorry. So we call him the father of taxonomy. Why? Because taxonomy is classification. Okay? So he came up with our taxonomic system. So therefore, he's the father of taxonomy, which is the study of classification. What is their taxonomic system? What is that? It is a system for naming plants and animals. And also grouping similar organisms together. Alright, so that is what our taxonomic system is, and that's Carolus Linnaeus. So let's go back over here and understand a little bit more about classifying each category that we have. So he grouped similar organisms together. Okay, and when he did that, he created a category, also known as a taxon. There's a number of taxons. First of which we wanted to look at is domains. Domains are your what we call largest taxon. Now, why are they the largest taxons? Because you'll have the largest number of organisms because it's the broadest category. Okay, largest or broadest. Maybe broadest might be a better word to say. Alright, so we have domains. Then under domains you have kingdoms. Yeah, let me erase that. Okay, now kingdoms, bacteria just kind of bounce down there. in archaea. And then your kingdoms of eukarya are plant, obviously, because that's what Carolus Linnaeus first came up with, is Plantae and Animalia. Okay, remember Latin terms. Excuse me. So we have plants and animals. And then we all of a sudden discovered, hey, there's these organisms that look like a plant because they kind of stick up out of the ground, but they're not. We learned a whole lot about them, and we learned they're in a totally different category altogether. So we created one called fungi later on. Then we discovered there's these unicellular organisms, and some of them have plant-like characteristics, others have animal-like characteristics, and then some even have what we would call fungus-like characteristics. But they have a nucleus, so they don't belong with bacteria. However, we got to find some place to put them, so we put them all together in a category or a classification system in their kingdom known as protist. All right, so protist can be animal-like, or they can be plant-like, or they can be what we call fungus-like. And I'm going to leave the fungus-like alone because this semester you won't ever really study about fungus-like, so we can just kind of keep going. Protozoan is what we call a animal-like protist. Algae, and I really hate that we say they're algae, but algae is what we call a plant-like protist. All right, so now we've covered our domains and our kingdoms. And let's talk a little bit about classification categories. So we have two types of cells. Remember, don't forget, you have eukaryotic cells and you have prokaryotic cells. And those are going to get classified in a domain and then also in kingdoms and things of that nature. I'm going to kind of go into more detail about that as far as your classifications go. Let me go ahead in just one second. Let me get over to chapter 4. Okay, and find where I want to go. All right, so chapter 4, we're going to skip over microscopes for now. Skip over staining for now. There we go, Linnaeus and taxonomic categories. All right, so he's also the one that allowed us to discover species and things of that nature. So let me see what else I need to study and cover here. Domains, there's domains. Testing. All right. So, I'm going to give you a mnemonic device to remember your different taxonomic categories. And a lot of people have learned this in a number of different ways. So this is your domains. This is your kingdom, phylum, class, order, family, genus, and species. Now, most of these categories, each one is a taxonomic category, or what we call a taxon. Okay, so each of these is a taxonomic category. It's very important to understand that we have certain characteristics that help us group organisms into each of these categories. So that way we can get down to the point of a specific organism or a species. I shouldn't say species anymore. I'm still so used to saying that. Let me back that up. Because that was changed not too terribly long ago to specific, but I can't spell. Still can't spell today. Hasn't changed. All right. Specific epithet. These two together gives us what we call our species name. Okay, so for example, we are Homo sapiens. Okay, that's our genus. And then our specific epithet is sapien. Together, that makes, and they usually capitalize, and you can just use the first letter. And then this is always lowercase. Sorry, it doesn't really look that way. And that's species name. All right. This is a two-word system that was created by Carolus Linnaeus. And we call this binomial nomenclature. Now, what is binomial nomenclature? Well, binomial, two words. And nomenclature is nothing more than vocabulary, typical words. Okay, your nomenclature. So, it is a two-word name that gives us our specific species, and it is consisting of our genus group and our specific epithet. These together give you your species name. And he came up with that 300 or 400 years ago. But understanding, let me get back to our taxonomic categories. Understanding how we group these will help us understand how we derive at our species name once we actually get to that point. So for example, let me see, I know your textbook has an example of how we classify. Yes, right here on page 114. And how it goes from Eukarya, that's our domain, to the kingdom Animalia. Well, Animalia is certain, they have to have a vertebrae, things of that nature. There's a lot of things that are classified there. And then your phylum, this one happens to be Arthropoda. So this is telling me it's a joint-legged organism, so therefore it can't be a vertebrate. Okay, it cannot have any backbone, anything of that nature, because it's not a chordate. Okay, a chordata or chordate is going to be something that has to have a vertebrae. So we understand that this is not going to be a worm or anything of that nature because it's got segmented legs. So therefore it has to be starting to limit the organisms that it can be in. Now we go to class. Well, this one is Arachnidae, which means that it is going to have a certain number of legs. You notice arachnids always have 8 legs. That's the rule. Spiders have 8 legs. And their bodies are always going to be segmented in the same manner. So therefore they are grouped in the same class. Now order. This one we get even more specific. Parasitiformis, which is your mites and ticks section. Now we'll get even more specific because you have what's called soft ticks and you have what's called hard ticks. Well, Ixodid is your hard tick. And then we have your specific genus and species, which is ixodes. Okay, so we have ixodids, and then ixodes and scapularis. I scapularis is specifically your deer tick, so that's your species. So you'll notice how this goes through a level of elimination, so to speak, to get to your main specific species. We use this a lot of times when we are trying to classify organisms and identify them. So I'm going through this to kind of explain some other things that we may use, such as taxonomic keys. So, taxonomic keys are used by people that classify, or taxonomists. I wish I could make my light brighter. I don't think I can. I can turn it off. All right, so taxonomists are people that classify, also medical clinicians, you know what, I'll just forget that word. Researchers, things of that matter, all will go through and classify using a taxonomic key. We also call them dichotomous keys. Why? Because they'll typically have a yes or no. I'm sure you've probably seen these on Facebook and other things that you may have looked at. Gram-positive cells, for example, this is an example of a dichotomous key. Is it yes or is it a no? And then you can keep going through yes or no, yes or no, yes or no, until you get to a specific organism, because you've done a process of elimination. Similar thing that we do in creating those taxons and taxonomic categories is that we are going to go through a process of elimination until we have a specific species. So this one just goes through and gives an example of what you would do, and we call these dichotomous keys. Why? Because there's paired statements worded so that only one of the two choices applies to any particular organism. This is very important when we go through and try to identify bacteria, because a lot of times this is the main way we can do so. And we go through and do a number of testing to be able to identify them and make sure we understand what bacteria is what. Trying to make sure I haven't missed any of the taxonomic information, because I just kind of briefly go over those, but I do want you to make sure you understand domains and what they are. Now, one thing I do want to remind you all of whenever I talk about genus, because it's going to be mentioned a lot. If I say a genus, well, for example, Homo. Well, you know what? We'll do horses, because that one, that one is really cool, and that one's easy. All right, Equidae, or it's actually Equus. Equidae is going to be their family. Genus is Equus. So there's three specific epithets. Let me just put SE. Under genus, Equus equus, Equus cabias, and an Equus zebrus. Okay? Equus equus is donkey. These are small. A lot of times whenever you have a genus, you're going to have like 20 or 30 organisms within that genus. So this one's just easier. Equus cabias. Sorry, that's supposed to be a lowercase c, so just pretend like it is. And that's horse. My spelling's not all that great. My writing. If you were writing on this whenever it's not down in the down position for a tablet, you'd understand. Equus zebrus, which is zebra. You notice I didn't put mule, donkey, or anything of that nature together here. Why? Because those aren't species. They have to be able to produce fertile offspring, and a mule cannot produce offspring, nor can a donkey, or anything that is what we call a crossbreed, because they have been crossed between species. So they are not a true species. So the genus, so if we said genera, the genera Equus. The genera Equus has donkeys, horses, and zebras. So you'll hear us talk about genera and genus a lot when we talk about bacteria, because you notice, for example, staph. Okay? A lot of you know staph, Staphylococcus. Very common bacteria that tends to be opportunistic and cause a number of different types of skin infections. So Staphylococcus is very common, and along with, we already talked about Streptococcus. Now, is that a species? No, it is a genera. We have a huge number of, a large number of different types of Staphylococcus. And depending upon the type, as well as Streptococcus, will depend upon some of the specificities it has. So when we talk about our bacteria, we're going to talk about genera a lot. And when we talk about those genera, just remember, this is not the species. It is the genus, which is a little larger grouping. So that means you don't have one, you have, say, 20 or 30 that are going to be in that genera. So I just want to make sure we understand what a genus is. Very, very important. Because you're going to hear the word genera over and over, and I want you to be able to follow me. I also want you to be able to follow me when I say prokaryote. Prokaryote is a bacteria or an archaea organism. We're going to talk about those some more. I'm going to kind of briefly going back over some of the major concepts here. Eukaryotic organisms. A eukaryote has a nucleus. Prokaryotes do not have a nucleus. Okay? And we'll talk a little bit more about cell types in chapter 3. Because that's all about cells. Kingdoms. We have bacteria and archaea, but then we have plants, animals, fungus, and protists that are classified under eukarya. Who was the father of taxonomy? Carolus Linnaeus. Now, what are some of the ways that we can actually classify our bacteria? Because that's basically what we need to make sure we understand, is how to classify bacteria when we're dealing with this course. Because we're going to be dealing with a lot of prokaryotic organisms. They talk about some of them on starting with page 116 and 117. We have physical characteristics. Now I'm going to go through a little video. So what I'm going to do is actually, I'll just go ahead and try to put it in here in this video. Is look at physical characteristics such as the colonies. Based solely on an organism's morphology. When we say morphology, we mean shape. I don't know if you can see what I'm pointing at over here. Morphology, we mean nothing more than shape. Or the physical appearance. Sometimes we'll look at the physical appearance of the actual bacteria itself under the microscope using oil immersion. And sometimes we'll look at what we call the colony. Now, when we're looking at a colony, we're literally looking at what we've grown on an agar plate or a petri dish. Similar to how we were able to isolate organisms by Robert Koch. So he was able to view colonies and be able to look at their shapes. Here's the cool thing about bacteria. They grow specific. Their colony shape is specific to their species. So you'll notice one that has a very special colony shape, and I'm going to show you one in just a second. And you'll be able to identify that organism very quickly. So let's go over here to Google, my favorite thing. And I'm going to look at Proteus. Okay, Proteus mirabilis. This guy is really cool. I'm not, that's a decent picture. I want a really cool picture. There we go. So, right there he is. I know Proteus immediately if I see it growing in an agar plate. So, right here, they have it growing in this petri dish. And you'll see how it's making these rings. And these rings are literally part of the organism. This is called a swarm when it grows in this manner. There's very specific ways that organisms will grow, and this one's just a very special one. So I like to look at Proteus just because I think it's cool and it's fun to look at. And there's a little picture of Proteus mirabilis. It has lots of flagella if you look at the actual cell itself under the microscope. And it's a rod-shaped bacteria. Let me see if I can find some other cool ones here. It looks like they got some really neat stuff on here on this particular website that I just happened to pull up. Ooh, I like that. So it's like some different sizes, shapes, and things of that nature. Perfect. Okay, we have all sorts of different sizes and shapes. Okay, so I have found the classification here. Let me move all this stuff out of the way. Let's talk a little bit about how we classify organisms and go into that. So physical characteristics is one, and we're going to talk about this for a few minutes because it's very important to look at morphology, shape, and colony morphology to help identify your microorganisms. We have some main terms that we use for certain shapes, and you'll notice here in the textbook, I'm using the current edition of the textbook. I had to go and find an old edition online so we can use the e-text because I want to show you a video so that we can look at classifying there. So if you're looking at my screen, you'll notice I'm looking at the previous textbook, so do not go by those page numbers. However, the page numbers for the book that I have here underneath the document camera are correct, and they are in the current edition. So we have sphere-shaped, also known as coccus. And then we have rod-shaped, also known as bacillus. That is the genus that they would be in. And then we have spirillus, which is our spiral-shaped. I don't see them over there. But those are your three main shapes. And let's talk a little bit more about some of these other shapes. And let's take a look at my screen. So you'll notice here that we have typical prokaryotic morphologies, coccus, sphere-shaped, I shouldn't say cocoa. Sorry, you're going to be like, remember that from now on. Coccobacillus, which is your shape that's not rod-shaped, but it's not really sphere-shaped. It looks like it's between. And then you have your bacillus, which is the rod shape. Comma shape is vibrio. Spirillum and spirochete. You know, spirillum has more of a wavy pattern. Spirochete's more of a spiral pattern. Then we have those that can't make up their mind, so they're pleomorphic. Pleomorphic is going to have a number of different shapes, and there's really not any organized structure as far as those specific bacteria. Now, when we're talking about these shapes, we're talking about the actual cells themselves. Remember that. And they're star-shaped here. We will talk about endospores here a little bit later. I'm not going to talk about them now. And we will talk about the class, how they form spores in the next chapter. So I'm skipping over this. And let's go ahead. I'm going to go ahead and flip over here to chapter 11 so we can take a look. And you'll know what page number I am looking at with these things. All right, it's at page 325 that has the prokaryotic morphologies that are typical. That's page 325. I kind of bounce around all over the place when we're looking at classifying. All right, let me go back to, there we are. That's what I wanted, but I don't see it here. Probably passed right over it. There it is. I passed right over it. Page 328 has arrangements of cocci and arrangements of bacilli. So you'll notice they will grow and they will form specific arrangements, and they look a certain way when you view them underneath the microscope, depending upon the species. So depending upon the plane of division, whenever they actually grow and divide, will depend upon how they are viewed. So there are certain types of bacteria that only grow in pairs of spheres. So therefore they're called diplo, meaning two, cocci. Some of them will grow in chains. Those are streptococci. Some of them grow in tetrads. Some of them grow in what we call a sarcinae, which is a group of 8. And then some have a clustering arrangement that looks like grapes. And those are staphylococci. These are all describing a number of arrangements of cocci. You notice they have 1, 2, 3, 4 different planes here that they can grow in, that they can divide in. I shouldn't say grow in, divide in. Here's bacillus. We can have 2 together, diplobacilli. Sometimes they form chains known as streptobacilli. And then sometimes they'll snap and form what we call a palisade. It's a V shape that they do. You notice they have not as many different planes of division with bacilli. So there's not as many different types of bacilli as far as your structural arrangements. So let's take a look at hopefully I'll be able to find this video. Well, I cannot seem to find the video, but I know I do have one already recorded. So I will go ahead and put that in as a separate video for you all to watch over how the different arrangements are and the different types of morphology of cells with bacteria. So let me go back to chapter 4. So we're bouncing back and forth here. Let's go back to chapter 4 and look at some other ways to classify organisms. All right, so we have shapes. We also have what's called biochemical test. Biochemical tests can be a number of different things. For example, this one we're growing bacteria in a test tube with a little test tube on the inside known as a Durham tube, and we put it upside down, and if we notice that gas starts to form as they're growing, well, that bacteria has to be a gas forming bacteria. Not all bacteria do that, so now we've eliminated a good number of bacteria from that process. Also, another thing we can look at is, do they cause a change in the color of whatever we're growing them in? Okay, that's going to give us another idea of what they're doing, because it may give us a presence of a particular metabolic reaction that causes the color to change. These are usually indicators that tell us it's there. Simple as we can identify that, something like this. Most testing like this is similar to I was about to go blood test, but that's a serological test. Similar to nasal swabs and things of that nature that we may run to see if we've had the flu or things of that nature. I don't know about the COVID test, or else I'd talk about that one, but I don't really know exactly how that one works. Serological test, that's similar to a blood typing test. So if you've ever run blood typing test on yourself to look for A, B, or O blood types, that's a serological test, specifically an agglutination test or antiserum. It's going to cause a change in the actual bacteria. So for example, clumping, no clumping, they're showing it here, negative result versus a positive result. Very similar to what you might do whenever, like I said, whenever you are actually doing blood tests. And most of you have done blood tests, so you can relate to serological tests in that nature. Phage typing. Here's a really cool thing. We have bacteria that eats viruses. Okay, so certain types of bacteria are going to infect viruses and destroy. I can't believe I got that backwards. I am very stressed out because I can't find my video. I spent way too long looking for it. Bacteriophage is a virus that infects and destroys bacteria cells. So just erase what I just said a moment ago. It is a virus that infects and destroys bacterial cells. I don't know why I kept saying that, kept thinking that's not right. Okay, so it is what we call a bacteriophage, or simply phages. So it is an eater. Phage means to eat. Bacteria eater. Okay, think of it that way. So if you release these bacteriophages and it destroys all that bacteria, well, it only eats specific bacteria. So that way you would know that it has to be bacteria X. Okay, it can't be anything else, because that's what it's telling you by identification of that. So that's one way. I'm not going to look at the other two types that they have here, but those first three are very, very common, and they show phage typing, what it would look like. See those clear little areas? Well, that means that all the bacteria on page 118, that means all the bacteria has been eaten out of that area. So that means that whatever whatever bacteriophage they released onto that bacteria that was growing here, whole thing, this was very effective. Okay. Whereas this one was not affected at all. So obviously, it's not that type of bacteria that is eaten by that specific bacteriophage. Okay, so now let's take a look at staining. Okay, preparing microscopes for specimens. I probably need to just stop. All right, preparing specimens for staining. Okay, staining simply means coloring specimens with stains, which are also called dyes. So last week, with the introduction to microscope, I had you guys go through and perform a staining procedure. So you went and did that. And it was a simple stain. It didn't matter which stain you were using. A simple stain is basically the procedure that you were doing, which is nothing more than one stain on the organism. Now, when we do this with the bacteria, we have to first prepare what we call a smear. Okay? So there is step-by-step procedure to create a smear. And we'll talk about those whenever we go into our actual lab that does staining. However, I do want us to understand what a smear is. It's basically bacteria and water. It's been a very long week. I know you guys probably feel the same way. Dried on your slide. And when I say slide, I mean a microscope slide, right? So basically, here's my slide, and I use wax pencil. Okay, and the reason why we use wax pencil to go around here, I'm gonna make up my two circles. I'm gonna do it just like we would do it in lab. Okay. And we use wax pencil for our circles. Now the reason why we use a wax pencil rather than a Sharpie or anything like that is because we want oil or wax. And that's going to allow, when we add water, the water is going to stay inside there. So it creates a well. And then we're going to take bacteria . And that changed over from a whiteboard? Sorry. And then we're just going to kind of swish it around inside the drop of water, inside the well, the circle. And then you let it dry. And it'll dry kind of a crusty white looking smear. So now we have what's called a bacterial smear. So now we have our bacterial smear, okay? And then we're going to have to do what we call fixing it, okay? Okay, so what are we doing when we fix our bacteria? It's called heat fixation. So basically you take a Bunsen burner. Let me see if I can find something online about that. Okay, so I did find a good video that goes over bacteria slides and heat fixing them. And the whole reasons for heat fixing, it goes over a couple, but we're going to go over 3 reasons because those are the 3 reasons that are in your textbook, and it's also in your labs. So we'll talk about that as well. I'm going to go ahead and push play on this video for heat fixing slides.
These are 4 slides that have just been prepared but not heat fixed. This top one is a little too thick, but it is fairly even. It could probably be a workable smear. This second one is a little bit lighter, but it's less even than the one above it. And you'll notice it's still drying a little bit, not quite ready to heat fix. You have to let the slide be completely dry before you heat fix, or you could cause a little droplet of water with live bacteria in it to go up into the air. That's called an aerosol. Someone could inhale that and get, for example, Staph aureus in their lungs, which would not be good. This third slide looks like it's about dry, although I don't see any bacteria on it. That can happen sometimes when you try to not pick up very much bacteria for the smear, and it actually ends up not transferring to the slide at all. This last one is, some things about it are nice. It's a nice thickness, but again, it doesn't have the even consistency that we look for in a perfect smear. But most of these are workable. Again, like I said, I don't think this one would be good to use though, because doesn't seem to have much of anything on it. So now let me show you how you would heat fix your slide. You hold it like this. The purpose of heat fixing is twofold. Number 1, it kills the bacteria so that they aren't going to make anyone sick. And number 2, like it sounds, it fixes the bacteria to the slide. They're essentially glued on to the slide after this process. I'm going to swish it through the flame 3 times at the very top of the cone. 1, 2, 3. That's all it takes. It's heat fixed. And then this one is dry now. I guess there's just missing bacteria in that one part. So 1, 2, 3, heat fixed. I'm still going to heat fix this slide even though I can't see. Oh, actually I can. Can you see that? There is a little bit of bacteria on there. This is very workable. In fact, this might be my best one. 1, 2, 3, heat fixed. And then the last one. 1, 2, 3. So now they're heat fixed. I can put them in my slide box if I want. If I decide they're no good, I don't even want to bother gram staining them. I might put them over on the waste cart. But a lot of times when you're beginning, you should gram stain all of them until you can get a feel for what makes a good slide and what makes a bad slide.
Okay, so she is using what the flame is coming from is what we call a Bunsen burner to go through and heat fix her slides, and she gave 2 good reasons why we would heat fix. For one thing, we want to kill the bacteria. We definitely want to do that. Okay, so we look here, smear is air dried completely. Why do we want to air dry it completely? Well, we don't want to have an aerosol being released through the air of the bacteria, because we can make someone sick and we don't want to run that risk. And we attach or fix it to the surface. Okay, so heat fixation is literally fixing it. Robert Koch's the one that came up with this. So those of you that were interested in Robert Koch, he did this as well. And discovered and was the first one to do heat fixation. We want to make sure that we kill the organism. We attach it, and there it is. Okay, fixation kills the microorganism, attaches them firmly to the slide, and then preserves their shape and size. So they'll coagulate and stick to the slide, and it makes it easier to stain them. So there's your reasons right there on page 106. I'm hoping you guys can see this well, because I can't see what I'm looking at. Principles of staining. Now let's take a look at some of the different types of stains and things of that nature that we use. Simple stains. Let me back up. Principles of staining, and we have different types of dyes. And I always get these backwards, so let's take a look at the two types on page 108, so that way you guys can correct me if you want to. Acidic dyes. All right. We have two main types of dyes, and we have basic dyes. Okay, acidic dyes, because they stain alkaline structures and work best in acidic environments. They are positively charged. They are negatively charged. Positively charged cationic chromophores are basic dyes. They combine with and stain acidic structures, and they work best under basic conditions. Okay, basic dyes are used more commonly than acidic dyes. Okay, acidic dyes are what we call negatively charged. Basic dyes are positive charged. Okay, so basic is positive charge. Acidic is a negative charge. This is important when we look at what we're doing with our dyes. Examples of basic dyes that are positive, and they are anything else I want to talk about? Acidic, yeah, dyes, basic dyes. All right, they are positive. Examples of a basic dye would be crystal violet, saffronin. We'll work with that one this semester. Some more that you used the other day. You may have used methylene blue, malachite green, even iodine would be classified underneath here. I mean, we don't typically use it as a dye, but it can be considered a dye, and it would be a basic dye. All right, negative charge dyes. An example of that would be nigrosin. When we will use nigrosin with our negative dyes, a negative stain. So here's the thing. Here's my cell. And does anybody remember what charge that the cell wall, I shouldn't say cell wall, excuse me, cell membrane. Told you I needed to stop while I was ahead earlier. The cell membrane has. It always has a negative charge, right? So whenever I put, let's just think logically. When I put a negative dye onto this organism, what's going to want to happen with the charge? If you said retract, you are correct. They're going to want to repel each other. So therefore, the dye will not stain the actual organism. Instead, it stains the outside. Let me get a different color. Excuse me. So it stains the outside of the organism, so we can see outer cell structures rather than the actual internal component of the cell. Whereas a basic dye, so here's my cell membrane here, and it's still negative, right? But it's positive, right? So a positive and a negative will attract. So therefore, the positive attracts to the negative, and it will stain the organism. And the rest it flushes off. Okay? So you'll see a nice depiction of the actual organism. And most of the dyes we've used so far do that. That's the type of stain they are. So we have two different types of dyes, acidic and basic. Remember the difference between one is positive, one is negative. Okay? Why do we want to look at the negative versus the positive? Why is that important? Well, the negative ones are going to stain differently than the positive. Let's make sure we understand that and we have a good firm grip on that. Now let's look at the main types of stains. We have simple stains, differential stains. These are two main types of stains. Your simple stain we did the other day. Simple stains are a single basic dye. Okay, crystal violet, safranin, methylene blue, all of these classify as a simple stain. Okay, crystal violet, safranin, methylene blue, malachite green, all of those classify as a simple stain. Simple enough, all you will do is just literally soak your smear and rinse it off and let it dry and view it under the microscope, and you are done. We go ahead and pause this for a second. Okay, so now what I'm going to do is just go ahead and show a video over simple stain technique so we can see someone performing the simple stain, so it helps us to understand a little bit more, because theory is one thing. When I'm sitting here talking to you about it, I like for you to be able to visually see it as well, and then that way we can keep moving forward with a nice firm understanding of what we're learning. So this is a simple stain technique that this man is going through, and he's doing a very good job of explaining these different steps and procedures and what type of stain we're using and why we're using it. So I really like this video.
Hello, in this video we're going to do the simple stain technique. Now first let's talk about the purpose of the simple stain technique. You see, bacteria don't have any color within their cytoplasm, so it makes it very difficult to see them under the microscope. So what we want to do is we want to impart color into the cytoplasm so we can clearly see the bacterial cells and all the structures within them. Let me go over the materials and equipment you're going to need. First of all, you'll need a bottle of distilled or deionized water. You'll also need a book of bibulous paper, a staining tray, a bacterial smear, which if you don't remember how to do a bacterial smear, go in and review that video, and finally an acid-base stain. Now let me talk about why we use an acid-base stain. You see, the cytoplasm of the bacterium carries a negative charge. Acids carry a positive charge. So when you put that positively charged acid-base stain on that negative cytoplasm, it's attracted to it, it bonds to it, and we get really nice contrast. Now in this video we're going to use crystal violet, but also what's commonly used is safranin, crystal violet. Any acid-base stain will work. So let's look at the technique. First, take your bacterial smear and put it on your staining tray. Next, take your acid-base stain and apply 2 to 3 drops onto the bacterial smear. Now let me show you a close-up so you can see what it should look like at this point. Then, after it's penetrated for 1 minute, we rinse both sides of the slide. We want to make sure and get all that stain off of that slide. Finally, we're going to blot all the excess moisture. So take your bibulous paper, place your slide within it, and gently press on it. And you'll have to move the slide throughout the Bibulus paper 2 or 3 times to make sure that you get all the excess moisture. And there I have it. I have a properly prepared simple stain. Now let me show you a close-up of what your slide should look like when it's complete. Now in closing, I want to talk about a few mistakes you should avoid. Number 1, when you're blotting that slide, be very gentle. Don't apply too much pressure. It's very easy to break the slide when you're doing this. I've seen it many, many times. Second mistake is don't rub the Bibulus paper or move the slide about within the Bibulus paper, because you can actually rub off the bacterial smear. And finally, and this doesn't happen very often, but I have seen it occur once or twice, make sure you're rinsing with distilled or deionized water. If you accidentally pick up your alcohol bottle, you'll decolorize your bacterial smear and essentially wash the stain right out of it. So there you have it. That is the simple stain technique.
I think that's a very good example of the simple stain technique. Let's go ahead and take a look at gram stain. Excuse me, let me back up and say differential stain. Most stains that we use in microbiology, we have to tell the difference between one type of organism and another. So we are going to go with a type of stain that we know as a differential stain. This means it uses more than one dye so that different cells, chemicals, or structures can be distinguished when microscopically examined. So therefore, we are able to identify specific things such as gram-negative versus gram-positive in our gram stain. This is our most popular staining procedure. Hans Christian Gram was the one that came up with it, so that we could use this as the most frequently used differential stain to identify between 2 large groups of microorganisms. Purple stain, gram-positive, pink staining, gram-negative. These cells differ significantly in their chemical and physical structures of their cell walls. So that is how we can be able to stain them and then be able to rinse, go through and stain again, and be able to identify between one type of cell and another. So let me see our steps here. Gram staining, it goes through the steps 1, 2, 3, 4 here. Flooding the smear with a basic dye known as crystal violet. That's how we get our purple gram-positive bacteria. That is known as our primary stain. It will color every cell. Makes sense, because we have just literally poured it over the top, just like he did in the simple stain. Then you rinse it. Flood the smear with iodine. Iodine is known as what we call a mordant in this sense, because it's going to set the original stain, okay, or our primary stain. It sets it kind of like whenever you wash your clothes and then you dry them in the dryer, and if anything was on them like blood, it sets the stain. It no longer can be removed. Think of it that way. Mordant, it sets the stain. It binds to a dye and makes it less soluble. Okay, now all cells remain purple. Now we're going to rinse them off with ethanol. It's just alcohol. Okay, only for about 5 seconds. This one says 10 to 30, 5 seconds. This is known as a decolorizer. Why? Because it's going to decolorize your gram-positive bacterium. Now, the only cells that should be purple are your gram-positive, and the ones that are gram-negative should be colorless. The reason why is because gram-positive cells have a thicker cell wall. Okay? So it's going to be harder to rinse the dye out. We're not going to talk about the cell wall structure until chapter 3. Now we have colorless gram-negative cells. We need to color them or else we cannot see them when we view them through the microscope. So we will flood it again with another dye. This one is known as our counter stain because it counters the primary stain. So therefore it's going to be our second stain to put on, and it's safranin. Now we'll go through and rinse this when we're done and blot it with Bibulus paper like he showed in the simple stain. Let it dry and view. Ideally, you should see both purple and pink bacteria. There it is right there. Both purple and pink bacterium. And that way you know you have gram-positive and gram-negative bacterium there. That is if you have gram-positive and gram-negative. Now, excuse me, there are some special bacterium that are very sensitive, and they cannot use typical acidic or basic dyes. So we use a stain known as acid-fast stain for these bacterium, and they are Mycobacterium and Nocardia. Those are the two types that use an acid-fast stain. I'm not going to talk very much about an acid-fast stain, but it's also a differential stain. Endospore stains. This is a differential stain mainly so that we can identify the structure within a cell. Okay, and we're looking for what we call an endospore. And we'll talk about endospores here later. But basically, we're looking for an endospore. It's something very specific with certain types of bacteria, specifically Bacillus and Clostridium species, can form what we know as endospores. So we use those. I'm not going to talk a lot about histological stains. However, we do have a special stain I do want to mention, known as the negative stain. It's also known as a capsule stain. This will be a negative stain because remember it is using a negative dye or a acidic dye. And remember those have a negative charge, and the cytoplasm and the cell membrane of a cell, of an organism, is going to be negative. Therefore a negative and a negative is going to repel one another, and it will not color the cell. Instead it will stain the background and leave the cells colorless. Eosin's another one. I mentioned nigrosin. These are acidic dyes that are used for negative staining. These are usually used to reveal the presence of negatively charged bacterial capsules. Okay, so we call them capsule stains. So it's looking at the outside of the cell rather than the inside of the cell. And you should see like a halo that surrounds the cell when it's encapsulated. There's also what we know as flagellar stains, where we focus on the flagella. I'm not going to really study about that or worry on that one. So those are your types of stains. Now we will go on and look at microscopes here in a little bit. I'm going to do a separate video for that just to clarify anything or any confusion you may have with those things with your paper. All right, so this is pretty much what we've talked about. Let me bounce over here. We talked about classification, our cell types, our domains that we have, as well as our kingdoms. We also talked about our taxonomic systems and the father of taxonomy. Our taxons, as well as our genus and species, as well and our specific epithet. Genus, we understand, is a general term for and could be for a group of organisms, not just one. We also understand what a smear is, how to dry our smears, how to fix our smears, and then the dyes that we will use in our different staining procedures. So that's pretty much chapter 4. If you have any questions, feel free to ask and let me know. I will also post a video over the cell structures and arrangements here in just a moment, whenever I can find the access to the microbiology e-text. I will definitely get that out there.